All right. Can everyone see my slides?
Yes, we can.
Yep, perfect. All right. So today I'm going to talk about protein structure prediction using quantum computers. And in particular, I will be highlighting like some parts of the two papers that we published on this. Both of these have been joint research projects with a research hospital here called Cleveland Clinic and IBM. So we have this like 10-year partnership to look into problems in biomedical sciences, healthcare and life sciences using advanced computing technologies such as quantum computing. And both of these were like the first project was funded by the US government here, and the second one was by an organization in the UK. Um, so let me just jump into it. I feel like most of the biological terminology is going to be familiar with the audience here, but let me just like recap some of it and how we can actually think about protein structure prediction in the context of quantum computing. Um, so as you know, like the proteins start in this like unfolded state. They're kind of like a necklace, right? Like they don't have like a particular shape, but to really perform their duties and like their functionalities within their environment, they fold into this like native state, which is the most like thermodynamically stable three-dimensional shape, sometimes referred to as the conformation or the conformer. And the goal of protein structure prediction problem is like if someone gives you this like unfolded like shape, the sequence of like amino acids or like the residues, I will use that interchangeably, can we devise these computational tools that are efficient, right? That can give us this like stable shape that we call the lowest energy conformer usually because it's the lowest energy. And of course, like in the cell or in the environment that the protein is in, like this happens in the scale of like very fast, right? Like almost like instantly the proteins like fold into their native shapes, and it's usually driven by this like hydrophobic effects between like the motor molecules, or sometimes it can be in a membrane, I guess, but it's driven by these like interactions. So the goal is to capture these interactions and like come up with like these computational tools that can mimic how the final shape is going to be. And if you look at like what kind of methods people are using in the field, what is the state of the art or SOTA, like in this, there are of course experimental methods such as X-ray crystallography, NMR, or cryo-EM. These are actually kind of the ground truth, right? Like people spend years in the lab, like verifying these structures and getting the final shape, which are quite important. It can be relevant for a disease, right? It can be relevant for a therapeutic, but those kind of like determine the ground truth. And there are also like this bucket of computational methods. Some of them are kind of like template based, and of course, I imagine most of you are familiar with like deep learning methods, classical methods such as like AlphaFold. When I was preparing these slides, like there was only AlphaFold 2, but we know that there is now AlphaFold 3. So these are kind of like trying to learn some structure, right? They don't necessarily see the physics or like the biology of the folding, like immediately, but they use like some pattern recognition. They learn, like they're trained on these like large data sets where the structural information is already available, and they try to sort of make predictions based on those patterns. And there is the other set of like algorithms that again, it's been I think part of the discussions also like in this workshop, that are kind of like more physics based, which are essentially trying to simulate like what's happening like in the actual physics of like folding, such as molecular dynamic simulations. And most of the quantum algorithms that, well, I'm going to talk about two work like that we've done. Both of them kind of go more towards I should say like physics based protein structure prediction. So there is no like learning, or there is no like training data. um, and for me, like one of the challenges, again, I'm not necessarily a computational biologist or a structural biologist, like, but it's an interesting problem for us. Like every time like we want to use this like advanced computing technologies, like one question is like, why? Like, what do we actually like need these technologies to tackle this problem? Um, and one of the largest like sequenced protein like databases, like this UnitProt, right? and these numbers are like a little outdated, but there are a little over like 250 million sequences there, where we have sometimes like partial information or full information, but for most of them, like we don't have their structural data. So the largest database in the world that has the structural information that are experimentally verified are, this is called PDB. You're probably familiar with this. And I think it's around 240,000 now. But if you look at it, we are kind of three magnitudes behind, right? It looks like the pace that we have, like we have this huge like database of sequence like proteins, but for large majority of them we don't have their structural information, and it looks like the sequences is becoming cheaper and cheaper, so we are able to sequence like more and more genomic like sequencing. We can do it much faster, but experimentally verifying these structures is like still very expensive and like time consuming. So certainly we need like better and faster and efficient like computational tools like to tackle this problem. So this is like in my mind, like if you think about complexity theory wise, like this is a scaling problem. Like we will never be able to match that in terms of how fast we can do it. And I think, again, most of you may be familiar with this, but the gold standard of structure prediction is CASP. So this is a blind competition that happens every 2 years, started in 1994. But this is a global kind of competition where hundreds of groups, different like research labs, they submit their predictions, right? And these are like blind targets. No one has seen them before. So even if you have a model that is trained on something, like, it doesn't have that structural information in it. And, as part of this, again, even though I'm not a computational biologist, like I spent a lot of time looking at CASP because I wanted to understand what is the state of the art, like what are people using like in this field. And this is also the place, where AlphaFold 2 actually became kind of like the breakthrough, and then later led to, one of the founders like receiving a Nobel Prize in chemistry. Like I'm sure you've seen it. But yeah, so like AlphaFold actually made its debut, and like it became like a big thing in the field. Like now everyone is using that, and it's a very, very powerful tool, right? And and when you look at like the top performing, I think this is from CASP 15 in 2022. If you look at the top performing, groups, folding like these groups, they all use like some variation of like AlphaFold 2. They do like minor tweaks to improve their performance. But the general message that I want you to take is like, in general, if you want to understand like what is the best performing tool, and like what targets are challenging, what kind of structures are like challenging for these, you look at CASP. Like this is one of the sort of the standards in the field. Again, I'm not from the field, so but like by reading into the literature, like these are the things that I've been seeing. Now going back to the quantum algorithms for protein structure prediction, there is like, if you look at, and the field kind of started in late 2000s, the first paper coming. And there are certain like commonalities, like if you look at these papers in terms of, now we have a completely not necessarily quantum native problem, right? We are not looking at like the molecular structures trying to simulate that. Rather, like this actually becomes more of a combinatorial optimization problem. Like if you map it, we have this exponentially growing, like configuration space. If you have like n amino acids, the number of possible configurations, the shapes that it can take is actually like a large space. It grows combinatorially. Then the question becomes like, can we find the actual like lowest energy conformer like in this large space configuration space? And in the field, like there are these like commonalities in terms of like mapping this problem onto a quantum computer so we can actually solve it, And usually it starts with like defining this lattice. So you can think of the lattice as like the integer points like on R3, right? Like the three-dimensional Euclidean space, or earlier papers actually start with the two-dimensional model. So, we know that this is not biologically like real because, I mean, I guess you can have like more planar, flattened structures, but most of the proteins are like three-dimensional, right? You cannot confine them to like 2 dimensions. And then you encode these either like by, encoding the turns that the protein is taking. You can think of it as like, I start at a point in R3 and I start to take a walk, and I just keep like hopping around like to the next lattice point and the next lattice point. So, but we need to find a way to encode this. Then you need to build a problem Hamiltonian, right, which kind of like describes the energy of the overall system. But of course, like we want to make sure that as we are like looking at these 3D conformations, we're not violating some geometric constraints. Like this has to be realistic. It has to sort of like capture the actual biology. And then in terms of like energies, of course, like when you have nearest neighborhood like interactions, you want to make sure that because of the interactions between these residues, like there's an energy, right, like free energy contribution that comes. And there are a couple models, but the most common one that, at least on the quantum side, people are using is this Pygmy-Jernigan model from 1996. And this helps you to score the conformers, right? If you have two amino acids and they interact, there's like this like negative energy contribution. This is essentially a matrix from which we pull the energies. And then mathematically, what we do is we essentially like sample these self-avoiding walks on this predefined lattice. And there are different methods, but the most common one is like then you can use this variational optimization scheme, like maybe you talked about it earlier, like variational quantum eigensolver or like QAOA. There are these like variational quantum optimization methods that allows you to find the lowest energy solution. And the field actually evolved like kind of as I mentioned. So we did like a very in-depth, like literature analysis. These are kind of like the big papers in the field, like from 2008 to like, this year, and it still continues to evolve. It's an active like, area. It goes from, more two-dimensional structures to three-dimensional structures, more efficient encoding, so you can get away with like, using less qubits, like to encode the same, protein sequence. There are different approaches. These are kind of like more technical, so I'm not going to get into that, but kind of like non-variational approaches, I feel like, Grover's algorithm, Grover's search proposals, but they are kind of hard to implement because, Grover's search is very expensive. We cannot implement it at scale now. The other one is this Kipu paper, like they have this approach called like bias field, digitized counter-diabatic quantum optimization, etc. So like it's a very rich field, with a lot of different approaches. And the earlier, like the very first paper that I showed, like in the earlier slides, is kind of using this tetrahedral lattice model. And essentially, in this like 3D grid, you have this like tetrahedral lattice that you build. Imagine like small pyramids like touching each other at the corners, and you decorate the entire like three-dimensional space with this. So those are going to be kind of like the corners of the pyramid are where you can actually have like the residues or the amino acids like sitting. And in particular, like you use a turn-based encoding, which means that for every possible turn that you can take, you want to associate with this bit strings. So that allows you to do that. And naively, the first thing you can do is for each direction, so you have 4 possible turns on a tetrahedral lattice. For each turn, you can say that, okay, like if I'm taking a turn in this particular direction, this is going to be 1 and everything else is going to be 0. So these are called like turn indicator functions. So in a way, like it's a function that returns you 1 if you're taking a turn in that direction. And for everything else, it's going to return like 0. Imagine like a piecewise defined function, like in math. And these allow you to sort of like encode the entire turn sequence. Imagine like you have the first amino acids, jump to the next one, jump to the next one, like following, the sort of like this tetrahedral like lattice points. And this allows you to encode the entire turn, but like in the language of like bit strings. And once you encode this turn, and you need to also think about how you're gonna encode the interactions. You can build a Hamiltonian. So I'm gonna briefly like talk about this again. I don't want to get into like too much technical details, but happy to talk about it later on if anyone is interested. But these first 2 terms are kind of geometry constraint terms. So this is called the growth constraint. So this essentially controls, as the lattice is growing, you're not gonna fold back onto yourself, or you don't have like 2 amino acids like occupying the same space. So it allows you to make sure that you have the correct geometry, right? Like you cannot have 2 amino acids like in the same space. Like that's not physical or like biological. And the second term is about more like side chains. It's the chirality term. So it enforces like you have the right chirality. But like these are all kind of like functions that you have to build, like to make up the Hamiltonian. And the last term is the interaction term. So like you compute like which terms are going to be, which residues or amino acids are going to be nearest neighbors. So you know like which energies are going to contribute to the overall fold. So this was kind of like the earlier, and this algorithm originally developed by the IBM Zurich team in 2021. And in our work, like what we did was like essentially take this algorithm, which was very resource efficient and really nice to work with, dig a little like deeper into this. And we actually wanted to go back and look at some CASP results to see like at which point like quantum is going to be useful. And this is a very busy like plot, but I will try to explain to the best of my ability. But in general, we know that as the protein sequences are getting larger and larger, the problem becomes harder, right? Like this is usually true. Like as the problem is getting bigger, problem usually gets harder. But we wanted to see also like what else contributes to the hardness of like this protein structure prediction problem. And this is kind of like how we redesign like or rethought about like the problem space and the parameters that makes it hard. And in particular, one axis, like you can think of it as the protein size. So as the protein size increases, the problem becomes harder. The other one is the number of point mutations. And these are all like analysis based on when we look at the CASP results and like try to sort of like systematically like define like what makes these problems hard, especially for the classical state of the art, which is the AlphaFold. So this is the number of point mutations, or you can think of it like how intrinsically disordered like a given protein is. So like in this scale, like, imagine as you further go along the axis, like the protein is becoming like more and more disordered, which contributes to the hardness. And the z-axis is like slightly more like technical, called the MSA depth, which is the multiple sequence alignment depth. And this is something they also like mention in their Nature paper when the AlphaFold first came out. And as if you don't have enough like a deep enough MSA, it's known that these models usually struggle. And in this work, like initial work, what we ended up doing was essentially carve out this like subspace of the larger problem space, where we can identify like these proteins, where we know that classically they're gonna be hard, so we know that like quantum has a chance to beat them, and this was quite interesting, and it gave us like a deeper understanding about like what are the classical tools that the practitioners of the field use, and at the same time like what are the inherent limitations of these tools. Again, this is a very busy plot, but I'm happy to talk about that later. If anyone has a particular question, these are all like CASP targets that we analyzed. And finally, also like we established this workflow, we wanted to test it on a small peptide. This is a very small sort of like the loop of a larger protein sequence, part of the Zika virus, and this is I think like maybe 7 amino acids. So what you see here is like the blue structure itself is the experimental loop. This is the catalytic loop of the protein, and then we benchmarked it against like different tools. The orange one that you see here is the result that we got from the quantum computer after we ran the algorithm. We took the same Hamiltonian and solved it like classically. Again, you can do this optimization classically as well. PepFold is another tool that people use, kind of simulating again like folding. And we also had the AlphaFold result, like to compare against. And what we saw was essentially the quantum computer actually gave us like very competitive results in terms of if you compute the RMST, right, which is the root mean square deviation. So it's a measure of like how far off you are from the experimental structure. And similarly, like, we also computed the radius of gyration, which is like how open and closed your structure is. It's a measure of like, like how well, like, the structures are sort of like opening and closing compared to each other. And we got like really good results from the quantum computer, definitely much better than AlphaFold 2, but again, this is a small peptide, so take it with a grain of salt. So that was kind of our first attempt, first like analysis, like looking into field and like running these things on an actual quantum computer. Then, as I showed in the earlier slides, we did like a very extensive literature research. We are like, okay, like this is where the field is, this is what people are using. what is next? Like how can we improve, the existing algorithms and the tools, like to perhaps like make it better. And one of the things that we wanted to do was essentially, as you're running these algorithms, if you remember, we had these like lattices, right? And the lattices like essentially limit what kind of geometries and what kind of structures you can represent. And the most advanced one, or like the most realistic one, like up until now, has been this tetrahedral lattice model, which only allows you to have like 4 possible turns at each lattice point. But it's been actually shown on the classical side that, face-centric cubic lattice, or FCC lattice, is actually much better, and people have been sort of experimenting with that since like 90s, and like no one has ever implemented the FCC lattice on the quantum side. And compared to tetrahedral lattice, FCC lattice allows you to have 12 possible turns, so it has like higher degrees of freedom. And it's also been to shown that it can give us much lower energies. It's a better fit in terms of like representing these structures. There is like this paper that shows it on these famous like Harvard instances of like 10 proteins, which is a benchmarking set people use. And more importantly for us, like people have been like showing, and we all wanted to verify ourselves as well, this lattice is actually better at capturing like secondary structures. So if you have like a alpha helical structure, like beta sheets, actually like FCC lattices can give like fit much better to these secondary structure. And what you see on the right side is like a bunch of different protein like PDB structures that we wanted to test and compare against like tetrahedral. These are all done classically, by the way. There is no quantum on this slide yet, but we wanted to take the native structure from PDB, fit the best FCC lattice, like how well it fits, fit the best like tetrahedral, and try to compute RMSTs, like if FCC is giving us like a much nicer fit. And you can see, like especially like look at the pink one and compared to the, like the green, and like the AlphaFold also is the orange one. And in most of the instances, like what we ended up observing was like FCC is actually fitting like really well, especially if you have these like alpha helical structures, like that are prominent, or like if you have this like beta sheets, kind of. It's actually giving us like really low RMSTs, and these are two other like benchmarking proteins like TripCage and like Paxilin people use. So this gives us like more motivation that we should go ahead and like encode this and build the algorithm for like running FCC lattice. And again, the rest, like the next couple of slides are like slightly more technical because it's on the quantum side, like how we do this encoding, how we build the Hamiltonian, and we ran into some issues like as we were building the Hamiltonian. It turned out to be like really expensive, like in terms of resources needed to run this algorithm. But we follow like this turn-based encoding. So if you think about like your amino acid, like this being the green sphere, now you have like 12 possible directions, that you can take. So if you're starting here, you can go like remain on the XY axis, right? Or you can move like vertically, you can go up or go down, and you still have like 4 lattice points. So when I say turns, like these are the 12 possible turns like that you can take at each point, and imagine this being like shifted like every time you move on R3. And there are some like nice tricks you can do, but because you have 12 directions, now you need to use like more qubits to encode this. So like each turn you need to spend like 4 qubits, or like these bit strings, like to encode the whole thing. And in total, like if you have n amino acids, we're gonna have to use like 4n minus 10 qubits. So like that's not great. Like that's still like linear cost, but like that's still expensive compared to the tetrahedral lattice model. And that's like kind of the trade-off. Like we wanna encode a richer geometry, and the price that we have to pay is, like we have to use like more qubits to encode the same structure. And there are some like tricks you can do, like these minuses and stuff like comes from, you can fix the first turn, and you can fix like 2 more qubits because there are some rotational like symmetries that you can exploit, to do this. But at the end, like, this is the total number of qubits to encode the turn sequence. Then we have to spend some extra qubits, so it becomes like slightly more expensive, like which is also the case for the other lattice model. But anyways, the Hamiltonian is quite similar to what you've seen earlier. So this is, we have essentially like two terms, H-back and H-overlap. Like these are kind of the geometry enforcing terms. This H-back is so the protein like doesn't jump to the next point and come back onto itself, right? So it doesn't fold back onto itself. And H overlap is like slightly more general. So you don't want like protein to just like jump around and come back to the same point and you have a clash like two amino acids like sitting on the same spot. So it avoids those kind of geometries. It like penalizes them. This redundancy is like we have extra unused qubits, so like it kind of gets rid of those. Like you can think of it that way. And the last term is the interaction, kind of similar to what we had before. If we have like two residues that are close enough, like amino acids close enough, and they interact, like we want to account for their energies. So this is like a pretty standard way of like building these kind of Hamiltonians that represents the overall energy. And again, I'm not gonna spend too much time, but this non-backtracking constraint, it uses these like turn indicator functions. And these turn indicator functions, like, the input is like a qubit-like sequence. And depending on like if the turn is in that particular direction, it's going to evaluate to 0. Otherwise, it's going to evaluate to 1. And you can use this like logical like Boolean operators like to build these like more complicated functions. Turn indicator functions, you can think of them as main building blocks. And then like, if you want to penalize like, folding back, for example, then that means, for example, this is the plus direction along the x-axis, plus direction along the y-axis. And then, you take that function, you put this, and function, like, and with like minus x minus y. So you actually like, have a function that represents going in one direction and coming back. So you can build these like really complicated functions like through these like logical like Boolean operations. But these essentially become the main building blocks for building the Hamiltonian. Similarly, we have the redundancy term. We have these like 4 sort of like basis vectors or like bit strings that we are not using. If you have 4 qubits, right, you have 2 to the 4 possibilities. So you actually have like 16, basis vectors, so to speak. But we only use like 12 of them because we have 12 terms. But we want to make sure that if these 4 unused ones show up, like we kind of penalize that. So this is just to take care of that. And then you also have the interaction term, again, picks up energies from the Miyazawa-Jernigan like matrix and uses that. But finally, the challenge for us was the non-overlapping constraint. And again, this really goes into like the technicalities of like when you have this kind of like combinatorial optimization problems, you usually have an objective function that you are trying to minimize or maximize, right? And in this case, because we want to find the lowest energy, we have an objective function, which is kind of our Hamiltonian, and we're trying to find the global minimum of that. Imagine if you took any calculus classes, right? Like you have this sort of like function that you define, and like you're trying to find the like the lowest value like on the y-axis, you can think. We're kind of trying to do something similar but much more complicated. But along with this function, we also have some constraints, right? Like you can think of it as the simplest case. We are trying to find the minimum of like a function f of x, but we also want x to be greater than or equal to 3, right? Like that's our constraint. We're looking for all solutions that satisfy that inequality. And we have a similar situation here, and those constraints now come from like these like geometries, right? Like for example, this is the distance function, and the distance cannot be like less than, it has to be larger than a certain value, because if the distance is like less than that, then it's essentially implies that 2 amino acids are occupying the same spot, so that cannot be a physical solution. So it's the same idea. We have an objective function where we're trying to find the minimum, but we also have this like geometric constraints that we represent in these form of like inequalities. And this turned out to be a very challenging task, and there's a standard way people do this in the literature called like slack variables. But we didn't want to do that because it introduces like extra qubits, and it becomes like really expensive, and it really limits the size of the proteins we can trade. So we wanted to look for ways like, that we can encode these constraints into our, problem without using slack variables, without using extra qubits, so we can still make this algorithm efficient. I'm not gonna again like go into like all the details, but we came up with like these two different methods to encode these like inequality constraints like without using slack variables. One of them is people usually refer to this as like unbalanced like penalization method, but we call this like polynomial fit. The idea is like very simple actually. So the inequality constraints, like these things that we are trying to encode, they look like these exponential decay functions. So they have a really high value. I don't know if you can see these like red dots, but around x is equal to 0, they have these really large values. Then they immediately like decay to 0 and they stabilize around that. And the idea is like we need to come up with a function. We need to fit a function there, right? If you've ever done any function fitting, that can sort of like fit nicely here. So we can penalize like these unwanted region, but for the correct like values, like we don't want to penalize them. And the naive thing you can do is like you can fit a polynomial here. But if you have like a lower degree polynomial, like a quadratic polynomial, degree 2, this is not going to look great. Like it's not going to fit well. And you can always measure this by something called like R square, which tells you like how well you're fitting. But there's a special like type of polynomials called like Chebyshev polynomials, which are essentially built from like these like sine and cosine functions. And we like decided to use that and using the least square method so we can have a really tight fit. And there's some technicalities like depending on how far like these two amino acids are, like you need to go higher degrees of like fit. But at the end, like we were able to do this. But the unfortunate thing is like this becomes very expensive very quickly. It works, like we showed that it works. But as you have larger and larger sequences and the residues, like the amino acids are like further away from each other, then you have to go to maybe like degree 50 polynomial, 60 polynomial, which makes it like very, very difficult to like encode this like, onto the quantum computer. The other method, again, I'm not going to get into like a lot of details of this, and maybe this like second picture is like more illustrative, is called like Lagrangian duality. And the main idea is, I'm a mathematician by training and like I'm a geometer and topologist, so I love pictures. The idea is actually, again, like very simple. Forget about like, the quantum. We're still doing like variational optimization, encoding everything on the quantum circuit and doing the optimization there. But in terms of like a, optimization method itself, which is also like exists in the classical world, remember like our goal was to find this like lowest energy, right? Like find the global like minimum of this function. And what Lagrangian duality, or in this case, like, this variational quantum eigensolver V-qubit constraint does is kind of splits this into like two problems. Instead of finding the minimum, we're trying to find the saddle point. And a saddle point, if you took like multivariable calculus classes, in one direction, it behaves like a minimum, right? So it's like a minimum here. And in the other direction, like it's like a maximum. So instead of like finding the minimum, we reformulate this in a way that in one direction we are looking for the minimum, right? And in the other direction we are looking for the maximum. So we kind of like split this problem using this like duality principle, and the Lagrangian is, you introduce these like extra sort of like Lagrangian parameters, but essentially we convert the problem into this sort of like saddle point search, like algorithm. And this actually like worked so much better in our case. And this is a common trick I think people use on classical like optimization problems as well. And of course there's a trade-off again, like you have to run this like multiple times. But it's like a well-defined problem. Like you start with your objective function and your constraints. You can build this like Lagrangian function and mathematically prove that it's equivalent to the original problem in this language of like Lagrangians now. And then you can take the dual function and you solve the dual problem. And there was like an earlier work that did this like in general for variational quantum optimization routines. And we adapted that to our particular problem and, wrote the code and like ran the whole thing. So, and finally, like, let me just show you some results and how this works. So we had like this 6 residue, pretty simple, protein, to be honest. But we just wanted to show that the new algorithm that we built works. This is a slightly bigger experiment, 24 qubits, and we ran this on the hardware. But what you see on this slide is actually the results that we get on the noiseless simulator. So in general, when you're running a quantum algorithm, it's usually like a very good idea to have a small like samples like size problem before you actually run it on an actual quantum computer, which is going to be noisy. You run it on a local, like on your laptop or like whatever on the cloud, on a noiseless like simulator, because on the hardware like there's going to be a lot of noise, right? Like so one of them is called like this matrix product state MPS simulator, which is like an efficient way, uses like tensor networks to simulate these. And these are two sampling results. So like let me just sort of like try to explain. So the one on the left is using the polynomial fit method. The other one is the Lagrangian duality or VQEC method. And the red line that you see here is sort of the probability of like getting the best solution, right? What we want is most of the probability accumulating like towards that like red line, right? That's lower energy. So on the x-axis, as you go to the left, the energies are getting lower, and on the y-axis is like the probability of like sampling that particular solution. And as you can see on the even on the noisy simulation VQEC, like this method is much more stable. It doesn't have a lot of like noise, and it gets like the top solution with really high probability. But in the other one, we did end up, because we're essentially doing an approximation, we're fitting these polynomials, right? So there's going to be some unwanted like solutions there. But still, it was able to recover the like one of the best solutions with high probability. And these are the hardware solutions. So we have two different generations of like quantum computers, like Eagle and Herons. And Herons are like our newer generation of quantum computers. They are much more like stable. They have like lower noise. They're actually like kind of much nicer to work with, even though they both use the same technology. Like Herons are kind of like the newer generation. So we wanted to see like if we were to run the exact same experiments on both of these devices, do we see any differences? And we certainly saw. So this is the polynomial fit method. The one on the left is the Eagle generation, and this is the Heron generation devices. And in both cases, again, like we were able to recover the top solution with high probabilities. And another like interesting observation is like we had these peaks, which like had higher energies, but they were still like that we sampled them with high probabilities. We wanted to see like if they are important. And when we computed like the radius of generation, like this is one peak, this is another peak, and this is another peak, we wanted to see the structures that correspond to these peaks and compute their radius of gyration. One interesting observation, and this is highly speculative, is that as we go from like higher energies to lower energies, like it starts like more of an like open, unfolded state, and slowly becomes like more and more compact. So it almost like mimics like this folding pathway that you see like on the natural landscape of protein folding. That was kind of interesting to see like they correspond to these, like peaks. So we start like a much more open structure, and these peaks essentially give us like more and more compact structures with higher probabilities, and the same thing on the right hand side, The other like technique, we did the same thing, like ran the whole algorithm on the two different generations of quantum computers. Again, like if you look at just like the raw probabilities, like we see much better results on the Kingston, but more importantly, I think like this method VQEC is much more stable compared to the other one. We see like a similar sort of like structure becoming like more and more compact if you look at these peaks, So that was definitely like interesting to see. And you can see, especially with this method on the right hand side, this is the Heron device. We don't have like that noisy peaks here, they are not prominent, but on the Cleveland device. And we used a lot of like, advanced like error mitigation and error suppression methods such as like poly twirling, like if you talked about it, or like dynamical decoupling. These are kind of like advanced like methods that allow you to like mitigate the noise in the system. So actually like your results are more reliable. But in both cases, we were able to like recover the results, And finally, we also wanted to see like how the probabilities like are accumulating, So these are like the energies. This is the cumulative probability, and what you want to see is most of the probability space is like in the negative energies, because the positive energies, they cannot correspond to any like realistic solutions, like because the energies like free energy should be negative in this case. So these are actually like. But one thing, of course, like you're gonna have these sort of like noisy solutions, but almost like 60 to 70% of the probability space was in the negative side. So that means that we are sampling actually from the correct part of the probability space. So I'm gonna just conclude by saying a few like remarks about the future directions and like some questions that I ask myself, and please, if you have any comments, I would love to hear. But it looks like especially like these lattice models that allows us to, map this problem onto quantum computers as a combinatorial optimization and like solve it. The classical sides seem to like move away from this like in the 90s, and they work with like this fully Cartesian coordinate mappings. The one question is like, should we do the same on the quantum side? If so, like, how do we actually like achieve that? It's not that obvious. And most of the classical tools, for protein structure prediction, such as now, like even like I'm sure if you've seen it, like even Apple has like a protein structure prediction tool now called like SimpleFold. All these companies, they have something like in the game, but all of them use like a very similar idea. They all like work with these, training like some model on this existing structural information, but the quantum doesn't do that. So is there an idea like that we can do the same thing on the quantum side as well and build like a, quantum like machine learning or like deep learning model on this? And for us, like people working on the quantum field, the most important question is there like quantum advantage, can we show that like we can solve a problem that is beyond like classical tools, that being like, MD simulations, like deep learning methods, like whatever people are using, right? That cannot be solved classically, but the quantum can give us either like faster or like more accurate results. And again, some follow-up questions like related to that, but I feel like that was a lot of information, so I'm gonna stop here. Please, like, if you have any questions, I'm happy to address them. But thank you for your patience, and thanks again the organizers for inviting me here. So yeah, thanks.
So do you have questions from the audience?
I'm having a little difficulty hearing, so feel free to like
Hello, can you hear me
ask the
clearly?
question. Yeah, I can hear this. Yeah.
First of all, I would like to thank you for your presentation. And secondly, we discussed the breakthrough of AlphaFold2, but it also failed to represent the structures of highly flexible, intrinsically disordered proteins.
Yeah.
Do you think that quantum computing models or quantum AI hybrid models could tackle this problem in the future? And if yes, how do you predict that it will solve this problem?
Yeah, no, that's definitely one thing. Like, as you can see on the slide, like even IDPs are like, even if you have intrinsically disordered like regions, that seems to be a major challenge for these AlphaFold type, that's what I call them, because they all come down to the same like deep learning architecture. They have this like transformer layers, like they feed into like this sort of like graph, like, inference problem. And they all use some type of like MSA information, it seems. I've seen some papers like, with this like language models and stuff, like now they also have protein language models. They seem to be like working better, and they seem to be not, they don't seem to suffer from like this MSA issue, but I don't fully know like how well they work for, intrinsically disordered proteins or regions. I definitely think like this is an area quantum can like bring in a lot of value. Like, to be completely honest, I'm not sure if this like combinatorial optimization route is the one that's going to give us the best way. I feel like to really like tackle the problem, we need to really capture like the chemistry and like the biology there, like do the things like at an atomic level. And we do have some like studies, hopefully that will come out soon, that tries to do that, right? Like go at a molecular level and try to simulate like the physics and like the actual like the chemistry there. I think that will be a very promising path. But regardless, I feel like because of the way like these deep learning models are designed, like I think they're inherently like incapable of capturing like these intrinsically disordered regions, because these are like regions are, they're not stable, like they just keep like they're dynamic, and the whole thing is dynamic. But it looks like we definitely need like a different approach. Again, these lattice models, they're good, but they also are very static. Like they assume that you have this alpha carbon, like you have the skeleton, right? Like the sort of like it doesn't see the side chains. So we need improvements like in these models as well. So like, if anyone's interested like working in this field and have ideas about like improving these models, it's definitely more than welcome. So, but yeah, that's definitely one direction that I feel like AlphaFold type, models like need help.
Thank you for your answer.
So I get a question too. Your model is very intriguing to me, and it's wonderful to see these results. But do you think it would be another interesting point to integrate variational method with perturbation method in your lattice model to increase some kind of the sampling that you may limit yourself? Would that be possible?
Yeah, I mean, so kind of like under the hood of like this variational optimization scheme, there is a perturbation that happens, but I think like it's not in the same way that we are thinking, because it's not like the perturbation of like the protein like geometry, but it's the perturbation of like usually you can think of it as like, we have this like energy landscape, right? Like for the protein, and like we're trying to find this like, like a global minimum, and the perturbation of the parameters like determine like which direction we're gonna go. So we keep perturbing like through the direction of like the gradient of like the function until we reach like this like global like minima. Sometimes it can get stuck in these like local minima, and that's why you see actually those like noisy, unwanted like, configurations. I do not know like from the top of my head like anyone that does like the actual perturbation of the geometry, but one thing I can tell you is, and this is something, my colleagues here who are, experts in like computational like biophysics, like they did this after the quantum prediction, we get this bare bone structure, right? But then like you need to do some like extra steps, such as like you include like the atoms, right? Like the side chains, and this is all done like post processing like classically. Then after that he also applies some like force field, like molecular force field, to relax the structure a little bit so it looks like more real. So this final structure, this orange one, like once we get the prediction from the quantum computer, it's kind of like this like sticks, like it doesn't have anything. But then there's a lot of like classical post-processing applied to like relax the structure and perturb it a little bit. So we did that like in both of the work like later, but maybe there's a way to do it like in the quantum optimization process itself. Like that's an angle that we didn't consider. So yeah, thank you for that.
Thank you very much. So I have also another question, maybe much more philosophical, but
Yeah.
So AlphaFold and all, they use the PDB database, and X-ray is limited to solid state experiments, and NMR can measure only small size proteins, and all are problem when it comes to proteins like intrinsic to the solid proteins or other cases. So the problem I'm encountering is I think quantum computing could be very useful for biological systems, since the biology is happening in a liquid phase usually, right?
Yeah, yeah.
So how difficult is it to add an implicit order model in your model?
Oh yeah, that's actually a great question. It's something like
Kenneth Max, Dr. Max, and IBM are working together, I think, in this direction.
yeah, I'm part of that project actually. Hopefully, I cannot say much like at the moment, unfortunately, until we publish the results. But we are looking at a protein structure, kind of much larger, but like really going into the chemistry. Like I say, is like through some like fragmentation methods. We are kind of like looking at it like atom by atom, and like through like a really large fragmentation. Like I'm talking about like hundreds of like like fragments that require like large scale like quantum computers, and also like high performance computing, like hours and hours of like running.
Yes.
It's hard, it's very expensive, but it's doable. So and yeah, in that project what we are also trying to do. So we both have the results for the gas phase, but also like implicit solvent effects. So that is definitely like a very interesting direction, because as you definitely like mentioned, those problems hard, and like the understanding the impact of the solvent is quite crucial, especially to capture the actual like chemistry. Um, so in this like particular more cartoonish, let's say, like model for protein structure prediction, I think there's a way to also like, implement that somehow. And I think that can actually help. This is like one thing that was always on my mind, but never had a chance to like really sit down and work on it. But I think that will be in particular helpful, because the moment you add like the solvent impacts, or like even the external like mean field like impacts on the protein's shape, then it opens up like these values for like how do we model like protein interactions, because you can think of like the other protein almost like the external like impact, or this like binding with like ligands. So it opens up all these like possibilities of like modeling these, but it's not easy, because then the model becomes like one has to be very careful like with the modeling of that. It looks like when we do this at the atomic level, there is a lot of like classical like chemistry tools that allows us to give us a good starting place. We can start with like this Hartree-Fock energies, like choose a basis, right? Like, and you start like modeling these interactions between like electrons, and like compute the energies. So that's what we're doing now for a protein, like for like two different like geometries essentially. So hopefully maybe in a couple of months like I will have more results on that, but that's a very promising direction that we want to pursue further, for sure.
Yeah, we would love to see your results actually in a couple
Yeah.
of months. Okay, thank you very much.
Thank you again. Yeah. I'm sorry, was there another question, or?
My question is, in effect, we are solving a nonlinear constraint equation with Lagrange multipliers and finding the constraint optimization problem. I was wondering, whether you had any chance to compare in terms of computational speed up with classical computational techniques, such as maybe gradient descent or other classical techniques, any computational speed up with respect to what we are doing using gate model.
So sorry, like the voice was a little bit low. So if I understood correctly, your question was about like how does this sort of like
computational speed up, whether with respect to classical computation versus gate-based optimization solution, any comparison, any computational speed up?
Yeah, no, that's a great question. And one that we should always like keep in mind, especially as we use like this variational. So like the way usually like VQE works is, I don't know if I have a picture here, but yeah, like maybe this is like this is a more complicated one, but so essentially we have a quantum circuit, and like we can choose it like however we want. Like usually we want to have something that maps like really nicely to hardware. We call these like hardware efficient ansatz, but that allows us to prepare like a quantum state. And then we have a Hamiltonian, which measures the sort of essentially allows us to compute the expectation values, like the energy of the system. So we sample with this quantum state and compute the expectation value, which is going to be our energy. Then this energy, like this circuit uses like these parameters, right? Like that we can kind of like variationally update. And the whole like VQE pipeline actually is like a hybrid algorithm that has a classical part, a classical optimizer, literally from like, SciPy, that you can get one of the classical like minimizers there. It can be like gradient free, or it can actually like use the gradients depending on that, like the optimization changes. There's like Kobila, SPSA, like, LFPGS or whatever. There's like a whole set of like classical optimizers there, and you can choose like whatever you want. Some of them work better. So for example, we know that like Kobila Yeah, and I think SPSA like work really well with the quantum optimization routines, and especially I think one of them is like gradient free. So like you only do these like local difference perturbations. So we did a lot of like kind of classical benchmarking to use like which classical optimizer is gonna work best because it's all also part of the quantum optimization routine because like that's the only way you compute the gradients, you decide like which direction you're gonna go, or you do the perturbations and you decide like which direction like you want to go next. Then you go back to the quantum circuit and update those parameters in the circuit, and then you do the whole thing again. So in most of the cases, and again I apologize, I don't have the pictures here. Maybe I can pull out like in a second, but it's really like a loop. Like you have the quantum circuit, you compute some stuff from there, the expectation values, feed them into the classical optimizer, and it tells you like sort of like how you should update the parameters of your circuit, and it feeds back into this loop. And what you want to see is like the energy going down until you have a convergence. So the classical optimizers are already part of this quantum optimization routine. In general, when I want to solve a problem like this, like fully like classically, I think I have it here. So we usually use like a classical solver, and actually at the scale of this problem, we probably didn't need it, but IBM has this like classical optimizer called like Cplex, which works like really well. And there is Groby, I think maybe like from Fujitsu, I don't remember. These are kind of like enterprise level, like really large scale. If you have like millions of like parameters, right? Like the problem is like very complex, you can still use these, but we had access to them, so we used them. So all the problems that I showed you, you can take the exact same like objective function and the constraint and very easily solve them. So none of these are sort of, if you were to map them as like classical optimization problems, they are not hard, they are easy. Which is a problem, of course. Like we want to get into a regime that we know that classically hard. But having said that, our motivation was, okay, like we can solve this using classical solvers, essentially classical optimization tools. But nobody really uses like classical, solvers for protein structure prediction because most people don't think about protein structure prediction as an optimization problem. So that's why we had the AlphaFold results as well, because if we are targeting like a domain-specific problem and we want to use quantum, what we need to compare against is like what is the classical state of the art. If we had like a logistic regression or like a graph-based like optimization problem, then we would compare against like classical solver, and that would be the benchmarking that we need to do like to beat. But in this case, we wanted to do it anyways because we know that like it's important to see like what are the limitations of this. But even like the solution that we got from the classical solver, like the Groby, it didn't give us like the same RMST. It had like a much like slightly larger RMST compared to the quantum solution. So I hope that like sort of answers your question, but it's definitely a very crucial point, like to understand, can we classically, like if we were to just think of it as a pure optimization problem, can we solve this,
Yeah, thank you. I mean, since you set up the problem, as an optimization, non-linear optimization problem, I was trying to compare it, with a classical optimization case, So I was wondering if you had any data, to compare speed up.
Yeah.
Accuracy we can see. Accuracy is close in both cases, but any numbers on speed up? Because we know speed up of optimization problems in quantum computers is not proven yet. We have speed up with Shor's algorithm, or Glover's algorithm.
Sure.
Exponential and square root of n, speed up. But in the case of optimization problems, we don't have any solid proof whether we have any speed up possible. That's the reason why I was wondering.
Yeah. No, that's a very important point, and maybe something to clarify, right? There are certain set of algorithms that we know complexity theory wise, like there is like a separation, exponential or like super polynomial, and the ones that you mentioned, like Grover's or like Shor's algorithm, those are like fault tolerant algorithms, right? We know that we need like at least like millions of qubits and a lot of like T gates and like a different kind of quantum computing architecture to be able to run them like reliably for a relevant size problem. And we know like how much speed up they provide because we know the classical solutions, like the complexity of the algorithm, and we can mathematically prove that, you know, either we get a quadratic speed up or like an exponential speed up, right? Which are great. For most optimization problems, we still have some complexity theoretic results, and things get like slightly more complicated here because the complexity classes themselves are not well understood. And if you look at like, you know, NP, P, like BQP, NP complete, right? All these classes, we have some instance like example problems. We also like, don't fully understand, you know, if one contains the other, etc. But the important thing here, I guess, is for most of the hard optimization problems, such as this one, if you map it, because this is an exponential like scaling problem, like you cannot get the exact solution, right? Like exact solvers are not gonna work because again, at a certain scale, what you have to do is get like a approximate solution, which means that now we are in the field of like heuristics. We are not talking about exact solutions, but approximate solutions. And when you talk about approximate solutions, the speed up or like this complexity analysis are very hard to make because now we're not doing like, you know, like this comparison in the language of like kind of complexity theory, you know, like big O notations and things like that. The same applies to this problem, right? We know that in terms of like space saving, like encoding the same like size problem, quantum gives us an advantage because like you can shrink the configuration space into something smaller, which is like the common trick people use. But to compare like the speed ups, you need to look at the clock time. Like you need to look at actually run the algorithms, right? Like and look at like how much it's taking, you know, in the classical optimizer. And in this case, I can guarantee you in these problems that I showed you, classical is going to be much faster. So in terms of like speed up here, it's hard to like quantify if there is like any benefit. But one thing we can say that as the problem space is getting larger and larger, there's going to be an inflection point where the classical optimizers are going to struggle and you have to use something else. Like at some point, the configuration space is going to be way too big that you're still using like approximate solvers and they're not going to work as well. Like you have to try something different, you know. So if you have like 100 amino acids, like no one's going to use these brute force like solvers because like it's not going to work. Like it's going to be way too expensive to run that. And the classical solvers such as Groby and the others, they're going to give you approximate solutions. So, you know, on the quantum side, like you also use like approximate solvers anyways. I hope that sort of gives you an idea about like how we sort of like look at these problems and how to sort of like compare the benefit of using quantum versus like classical solvers.
Thank you.
Yeah.
So, are there any other questions? I think this is it. There are no more questions. Thank you very much for your interesting talk. It was wonderful, and thank you for your time.
Thank you. Yeah, it was my pleasure. And anyone in the audience, like if they have any follow-up questions or like interest, you know, feel free to reach out to me. I'll be happy to talk about it.
There's a lot of interest, Hakan. So,
Sure.
yeah.
Okay.
Thank you.
Thank you.
Okay.
Thank you. Thank you so much again for the invitation. It was my pleasure. And yeah, thanks for a very successful and interesting event. Yeah.
Thank you. Okay.
Bye.
Bye-bye.